Effect of SARS-CoV-2 virus S1 protein binding to ACE2 on ENaC regulators in cultured adult human fungiform, HBO, taste cells. COVID-19 often causes long-term loss of taste, smell, and chemical sensing. The virus's spike protein, specifically the S1 subunit, binds to a human protein called ACE2, which is part of a system that helps regulate blood pressure and salt balance. We think this binding disrupts the balance between two key pathways in this system, possibly affecting how taste cells sense salt. To test this, we studied human taste cells and found that they contain ACE2 along with other proteins involved in salt detection, including ENaC, a salt sensing channel, and TRPV1, a protein that responds to heat and spice. Our findings suggest that these proteins work together to influence how we taste salt. When we exposed taste cells to a modified version of the S1 protein, it reduced ACE2 levels, which could explain why COVID-19 alters taste. This research helps explain why COVID-19 affects taste and may lead to changes in salt preference. Understanding this mechanism could improve treatments for taste disorders caused by the virus. Introduction. Salty taste in humans is partly controlled by specialized proteins called epithelial sodium channels, ENaCs, which help detect sodium in food. Unlike mice and rats, humans have an extra ENaC component called the delta subunit, which may change how we perceive salt. These channels are influenced by hormones like aldosterone and estrogen, which can affect salt cravings, especially in women. Our study explored how human taste cells detect salt by examining whether the delta ENaC subunit works alongside other proteins, including hormone receptors, like GPER1, linked to estrogen, and a heat-sensitive protein, TRPV1, usually found in pain-sensing nerves. We also looked at whether components of the renin-angiotensin-aldosterone system, RAAS, a hormone system that regulates blood pressure and salt balance, are present in these taste cells. We think these proteins might work together in a complex to fine-tune our salt sensitivity. Additionally, since COVID-19 disrupts taste and smell, we investigated whether the virus's spike protein, S1, affects salt taste by binding to ACE2, a key RAAS protein in taste cells. We found that when a modified S1 protein binds to ACE2, it reduces ACE2 levels, which could shift the balance of RAAS pathways and alter salt taste perception. This research helps explain why hormonal changes, infections like COVID-19, and other factors might influence how we taste salt. Understanding these mechanisms could lead to better treatments for taste disorders or even help manage high-salt diets linked to health problems like hypertension. Conclusion. Our study reveals how human taste cells detect salt through specialized proteins called ENaCs, particularly the delta subunit found in certain taste cells. Unlike rodents, humans have unique salt-sensing mechanisms influenced by hormones and proteins like TRPV1, a heat spice sensor, and components of the RAAS system, which regulates blood pressure. We found that these elements interact in taste cells, forming a complex network that adjusts salt sensitivity. High salt conditions increased delta ENaC levels while decreasing TRPV1, whereas capsaicin, the spicy compound in chili peppers, had the opposite effect. This suggests that diet and sensory stimuli can directly alter how we perceive saltiness. Additionally, the COVID-19 spike protein, S1, reduced ACE2, a key protein in taste cells, potentially disrupting salt taste by unbalancing the RAAS system. These findings help explain why infections like COVID-19 can impair taste and how hormonal or dietary changes might influence salt cravings. Understanding these mechanisms could lead to better treatments for taste disorders or strategies to manage excessive salt intake, which is linked to hypertension and other health risks. This important article is authored by the following scientists. Thank you for watching this video. Please subscribe to our channel BP International to watch more videos in the fields of science, technology and medicine.